Sidney Brenner and the Francis Crick are the scientists who have won the Nobel Prize by working in different areas. So these scientists have demonstrated that the nucleotides in the DNA, they function in the form of triplets. Means that three nucleotides, consecutive nucleotides, they comprises a codon that is in the form of a triplet. And each triplet or codon codes for one amino acid in a particular protein. So these scientists, they were able to introduce the process of deletion within the DNA sequences. It means that they have deleted the particular nucleotide from the sequence of the genes. So they have demonstrated that when the one nucleotide is deleted, the protein has a very drastic effect that were transcribed from that gene. And if the two consecutive nucleotides are deleted, the protein that is synthesized from that gene is again drastically affected and its function is not properly functioned. So the third option is when they deleted the three consecutive nucleotides, then they have observed that the function of that protein is not much affected. So here we can see here that there is a phrase written here, the sly fox and the shy dog. So we can see that all of these words, they are written in the form of a triplets. Means that there are three alphabets in each word. So same is the case in the DNA. In the DNA, one consecutive three nucleotides comprises of the triplets, and it is also called the codons. So if one of the letters Y is removed from the word sly, so it means that Y is removed, then to make it the triplet form, F will move in the position of Y. When F will move here, the two letters will left with O and X. To make it the triplet, A will move after the X and so on. So after this shifting, the words we will get is the, we can read the, then afterwards, then it is created a very nonsense characters, we cannot recognize these. So if the two letters from the word sly is removed, Y and L. So again, if two letters are removed, then definitely then these two letters will move in place of Y, L and Y. So again, the letters that are generated, they have a no meaning. So in case if we remove the three letters consecutively SLY, then again we will come up with a very useful information. For example, if three letters are removed, then we will get the sentence as the fox and the shy dog. The fox and the shy dog. So it means that if we remove 1 and 2 alphabets, the arrangement will be disturbed. And if we remove the 3 alphabets, the only that codon or the triplet will be removed, the other information will remain intact. So same was the experiment of the Sidney Brenner and the Francis Collins. So after the discovery of the codons, another scientist, Charles Janowski, he come up with the experiments and he has proved that the gene and the corresponding protein of that gene, they both are in the collinear arrangement. It means that the first codon in the DNA is coded by the first amino acid, and the second codon, it codes the second amino acid of that protein. Similarly, the third codon will code the third amino acid and so on. So Janowski's experiments were so influential that no scientist has argued about the experimentation and about the nature of the codons, whether these codons are consecutively placed in as a stretch of DNA. So this theory was acceptable for the 2 decades after the discovery of this collinear nature. And most of the biologists, they thought that the genes or the DNA, they are composed of the codons that are comprised of the long strings. So after the discovery of the codons, and there is no argument about the codons, then after approximately 15-20 years, a new discovery was reported, that was the split genes. It was reported that humans, they have the split genes within their genomes. It means that the codons or the genes, they are not continuously stretched throughout the DNA. Rather, they are the stretches of small pieces intersected by the another type of DNA that is called the junk DNA. And this junk DNA do not code for any of the protein. Now we know that this junk DNA is basically also coded into a non-coding RNAs. And similarly, we know that human genome is much larger as compared to the prokaryotes, for example, the bacteria. And definitely it's not surprising, because the human genome is much more complex. So that is why it also bears the more genes as compared to bacteria. But rather surprising thing is that within the vertebrates or within the animals, higher animals or eukaryotes, there lies a disparity. For example, the salamander, that is an amphibian and is just like a lizard. Lizard is a reptile, but it just like a reptile, there is an animal named salamander. So it means that the salamander is not a very big animal. So the genome of the salamander is 10 times higher as compared to the human genome. So this disparity was removed with the discovery of the junk DNA. So we can assume that the genome of the salamander, it contains more junk DNA and less coding genes as compared to the humans. So we also know that the human genomes, in the human genome, there are present many genes, and these genes, they are short stretches of the nucleotides, and they are separated by the long stretches of the junk DNA, and we call these stretches small as exons and introns. So the exons, they are the coding part of the gene, and the introns, that is the junk DNA, is the non-coding part of the gene. We can also say that the analogous, that the genes that are in the form of a short stretches can be compared with an example of a magazine. For example, the split genes are analogous to a magazine article that starts from page 1, and then again it continues from the page 13, and then 43, 51, 74, 80, 91, and so on. And the pages that lies in between these pages are the advertising pages. So we can compare that the introns that are present between the two exons, they are the advertising pages, or the advertising introns that is present between the two exons. And the formula is very simple. For example, if there is a gene and it comprises of 4 exons, so we will minus 1 from that 4. So 4 minus 1 will be, the answer will be 3. So it means that 3 will be introns. So for example, if the gene has 7 exons, then the introns will be 7 minus 1 is equal to 6.